Welcome to lecture on protein structure prediction. Protein tertiary structure prediction. Protein structure prediction is the inference of the three-dimensional structure of the protein from its amino acid sequence, that is the prediction of its folding and its secondary and tertiary structure from its primary structure. Protein tertiary structure prediction is a research field which aims to create models and software tools able to predict the three-dimensional shape of protein molecules by describing the spatial disposition of each of its atoms starting from the sequence of its amino acids. Methods using in protein tertiary structure prediction. Most tertiary structure modeling methods such as Rosetta are optimized for modeling the tertiary structure of single protein domains. A step called domain parsing or domain boundary prediction is usually done first to split a protein into potential structural domains. As and when the rest of the protein structure prediction, this can be done comparatively from known structures or ab initio with the sequence only, usually by machine learning assisted by covariation. The structures for individual domains are docked together in a process called domain assembly to form the final tertiary structure. In computational biology, ab initio or de novo protein structure prediction refers to an algorithmic process by which protein tertiary structure is predicted from its amino acid primary sequence. Homology model building or threading. The most robust of the structure prediction techniques is homology model building or threading. The threading methods search for structures that have a similar fold without apparent sequence similarity. Threading takes a sequence with unknown structure and threads it through the coordinates of a target protein whose structure has been solved by X-ray crystallography or NMR. Although techniques such as threading are obviously very powerful, their current requirements in terms of both hardware and expertise may prove the obstacle to most biologists. In an attempt to lower the height of the barrier, easy-to-use programs have been developed to give an average biologist a good first approximation for comparative protein modeling. The use of Swiss model, a program that performs automated sequence structure comparisons, is a two-step process. The first approach mode is used to determine whether a sequence can be modeled at all. When the sequence is submitted, Swiss model compares it with the crystallographic database XPDB, and modeling is attempted only if there is a homologue in XPDB to the query protein. Another automated protein folding recognition method developed at UCLA incorporates predicted secondary structural information on the probe sequence in addition to the sequence-based matches to assign a probable protein fold to the query sequence. The second approach compares structures with structures in the same light of the Vector Alignment Search Tool or VAST. The VALI algorithm looks for similar contact patterns between two proteins, performs an optimization, and returns the best set of structure alignment solutions for those proteins. CPH Models is a protein homology modeling server. The template recognition is based on profile-profile alignment guided by secondary structure. This is how the primary protein sequence or primary structure, which has been sequenced by Maldito, can be converted to the specified file formats, and the three-dimensional structures will be modeled by using several servers like Swiss model. These are some of the website informations. Coming to the importance of protein structure prediction. If the protein structure is known, then one can predict its function on the basis of structural similarity. One can also predict which molecules or drugs can efficiently bind and how they will bind to the protein. This is the utmost importance for designing of a drug. Coming to conclusion, protein structure prediction is the interference of the three-dimensional structure of a protein from its amino acid sequence, that is the prediction of its folding and its secondary and tertiary structure from its primary structure. Protein structure prediction is one of the most important goals pursued by bioinformatics and theoretical chemistry. It is highly important in medicine, for example in drug design, and biotechnology, for example in design of novel enzymes. These are the references used for my presentation. Thank you.